Por su pesar de un nome es Barbosa, es de origen portugués, eu não falo português. Se eu tento de falar em português, eu falaria um portunhol, mas mais espanhol que Porto. Pero só em seguida eu acho que é melhor falar em inglês. So, thank you for coming. Thank you for coming. Thank you for to take the decision to be here and not on the beach. I try to demonstrate in the next hour that you have taken the best decision. And that I want to talk to you today is about the potential effect of the climate change of penguins, the physiology of the Antarctic penguins. And as all of you know, there is increasing temperatures in the last 50 years. On average, there's an increasing all the year, but there are some places, some regions in the earth that are more increasing the temperature than the others. Some of the places are located in the polar regions. But these increasing temperatures are not homogeneous in all the regions. The Arctic is more homogeneous and the effects are more deeper than in Antarctica. And in Antarctica, the region where the temperatures are increasing more is located in the Antarctic Peninsula region. It's more or less assumed that in the last 50 years the temperature has increased 2.5 Celsius degrees. And so this region that can be considered together with the Arctic, one of the places of the Earth where their temperatures have increased more and faster than any other place. This situation, this context gives us a good opportunity, unfortunately, but good opportunity for research. We are now at the same time that the climate is changing. It's not the first time that the climate has changed in the Earth. There are glaciation in the past. But we were not here over that time, so we have now the opportunity to know how this process is moving, is changing, and how the animals has to adapt or has to respond to these changes. The consequences of the temperature increase in the Antarctic Peninsula can be illustrated by the retreat of most of the glaciers in the region, in the area. More or less, the 87% of the glaciers have retreated in the last 50 years. There are some others that are advancing, but most of them are retreating. This is pictures of how this is a Palmer Station in the half of the Antarctic Peninsula, and you can see how in several decades the ice of the glacier has retreated in this area. It's more general for all the Antarctic Peninsula. Other things that happen in this region is this integration of the ice shelf. One of the more famous, because it was at the very beginning of the process, is the disintegration of part of the Larsen ice shelf in 2002. That it collapsed, and what it's also a new opportunity, unfortunately, but opportunity again, to study some of this area by the people that work with benefit organisms, because in the former time with the ice shelves, it was not possible to go there, to go under the water to see what happened there. It has been a very good opportunity for research. But obviously it's not a good thing. Another consequence of the temperature increase is the changes, the climate change, the changes in sea ice extension. As I mentioned before, the Antarctic continent is not homogeneously responding to the climate change. There are most of the container is cooling, and then there are some increase in the last decades of the CA extension. But again, there is a place in the Antarctic continent where the sea ice is decreasing. That's again in the Antarctic Peninsula. You can see in this graph how there is a clearly link between the increasing the air temperature and the loss of the sea ice system. And this is this increasing temperature and these different kind of things that are happening as consequence of the life, as consequence of the organisms. And one of the organisms that are responding in different changes, in different ways, are the penguins. Following the talk of my colleague, I think you have more information that I can give you now. But briefly, there is this species of Pygospherids that are responding in different ways. The adelie and the chinchlet are decreasing their population. This is data from the Stranger Point in the King George Island, and this is data from the Barbosa penguin rugby in the Station Island, from the chinchlet and in this case from adelie. But as Mike has mentioned this morning, the Gen 2 is increasing, has a stable population or even increasing. So these populational changes are not local. In this case, these are data for chinchlet penguin, and this is a regional pattern. You can see that most of the penguin colony, penguin rockeries in the South Shetland are declining. Some of them are disappearing. Some of them are very close to disappear. And on average, the decline of the population of the chinchlet penguin in the region is about 60%. So we have data, more or less good data about populational changes. It's clear that this kind of data is easier to get than other kind of data. So we go to the field, we count the penguins, we do this every year, and we have all the variation of population, and we obtain trends and patterns of the popular changes. But behind these populational changes, there are a lot of factors that can influence and that we have to try to find the mechanisms that explains these populational changes. And this is what we are trying to do in the last 10 years and in the future. So this is a graph that shows the potential effects of the climate change, and we can add all other factors on this final result that can be the populational changes. And this is what I will try to develop in the next minutes. This is, as I mentioned, what we are doing in the last 10 years in our project that is called PINGU. It's PINGU from Pinguino and from Clima in Spanish. And we worked a few slides about how we are working on this. We work with the three species of Pygospherids, the chinchlet, the Gen 2, and the adelie. We use three approaches. One approach is obviously working with climate changes at long term, so we are taking samples and measuring variables, different variables in different years. This is at this moment the years we have data. Now we have stopped for some years, and we will start again in a couple of 3 or 4 years, because we don't. We think that we don't need long-term, a continuous sampling of some variables, and it's better to have some small sampling of different years in the first time, and to do the same in the second time, and then we can cover without spending a lot of money. We can cover almost 15 years. We will have enough data to compare the first period with the second period. We do also a geographical approach because there is clear environmental differences in this geographical gradient. This is the three species, and these are the different populations we sampled at the moment. And as I mentioned, the environmental differences that we can find in this geographical gradient will allow us to to see how these variables we're interested in are changing, and this allows us to make prediction just in case temperatures will change, reflecting the difference that we are now in the temperatures between the north place of the Antarctic Peninsula and the southern place. And we have the other approach. This is the more deeper approach, using the facilities of for instance, the Spanish-based Gabriel de Castilla, the Argentina-based Carlini, and the Bioscam in the Livingstone Island. And in these places, we use experimental and observational studies that we have remained there for the 3 months, because I don't mention before, but the geographical and the long-term approach, we just spend some hours for sampling and we go to another place and to another place. But this is not enough for doing some of the more deeper study, and this is the reason we use the facilities of the bases of the stations and the camps that allow us to remain there for 3 months and follow all the visits of the day. So some pictures for illustrating the working process. So this is a modification, a recent modification of the graph we were seen before. It's more or more the same, but now I have presented this in the last SCAR conference in New Zealand. And we had some of the question of the things that we are now involved in our current projects, like the changes in our sea level spread bubbles, in relation with all the things. And we also had the effect of the human disturbance. And we can add here some of the things that Mike spoke this morning, the harvesting of the krill and different other factors that can more or less has the similar consequences of the climate change. So in the next minutes, I will develop all this graph showing how we are addressing the information on how we are disentangling this relationship between some variables and others. You will see at the end of the talk that there are a lot of work to do, so we are now at the beginning, but I hope that I can explain this clearly for you to understand. First, the climate change, the relation with the climate, the decline and tide change. I will have not use more time to explain this because I did it this morning very, very well. So, but just a few words. It has detected the decline in the krill abundance in the last 25 years. And this krill decline seems to be related with the reduction of the CAF, of course, always speaking in the Antarctic Peninsula. And the reason is because there is below the sea ice, there is the place where the phytoplankton and the microalgae is living. You can see ice core, and this color is represents the presence of the microalgae and the phytoplankton. And this microalgae and the phytoplankton is the food of the krill. So if we have less sea ice, then we will have less phytoplankton, and then so that the krill will decline. This is illustrated in this graph. This shows differences in the production of chlorophyll in the northern region of the Antarctic Peninsula in comparison with the the southern region of the peninsula, and you see that there is a clear decline of the production of chlorophyll that is indicating that the phytoplankton is declining. This is also show the loss of the reduction of the sea ice and the effects of chlorophyll. So there are some factors affecting the krill. You know, the krill is the central one of the central point of the Antarctic food web, and for instance, for the penguins is the main prey, at least in the last 300 years. But so what happened with the krill with affect the predators? For some data, it's been calculated that in South Shetland, the birds and the marine mammals eat 800,000 tons of krill in the year per summer. So you can imagine that the importance of the krill for all the animals, all the vertebrates at least in the Antarctic is very important, and some changes in this prey has consequences in the other organisms. So this is a repetition of the talk of this morning. It's possible to have evidence of the past of the changes of shift diet in the past using the excavation that Steve Fensley has done in the different parts of the Antarctic Peninsula and the Ross region. And he published a paper in Antarctic Science that showed that in the medieval warm period that happened around 1,000 years ago, the Adelie penguins changed their diet, consuming more squid than after in the Middle Age, which was more colder. That it more the main prey was a krill. So this has been happening in the past. So we can expect that it will happen. If this happens now, it will happen in the next future. If the temperature and the environment is continue changing. So sorry. So now we go to the next step. This diet changes can affect other organisms like the parasites and pathogens, mainly parasites, because if the penguins have to look for new prey or for change to other less often eaten prey like the fishes or the squid, they will be exposed to new pathogens, to new parasites. They will be to defend with the immune system. We will have problems if the immune system is not very well fit with these new parasites. So there will be effects of the population with the effects of the parasites. And on the other hand, there is a clear link between the climate change and the increase of temperatures and the increase of parasites and pathogen effects. It's clearly demonstrated that the increase of climate change or increase in temperature have affected the parasites, increasing the distribution range, also the abundance, and also the virulence of how the parasites and pathogens are interacting with the host. So we have to know what happened with this step. We have to know what parasite or which parasites or pathogen are present in the penguins, which are the effects, but we have little information of that. We did a review published in 2009 about the presence of the pathogen and parasites in the penguins. We revised 175 papers, and as conclusions, we found that the available information is incomplete in terms of host species. There are not many species studied, species of host. In terms of parasites, we know very little about the parasites and the pathogens and the geographical location. So sometimes, in former times, there were some researchers that went to a place, recorded the parasite were present there, but nobody continues the war, and we don't know after 20 years what happened with these parasites and the effects in this place. And at the same time, the research is focused on few places, and there are many different places where we don't know almost nothing about these interactions between the parasites and the hosts. So even when we have information, we have just information about the presence of parasites, we have almost nothing, no information about the prevalence of the parasitism intensity, so it's not possible to evaluate which is the importance of this parasite for the host. There are only 14 studies that was focused on physiology related to parasitism. That's important to know the effects. And there are just 4 studies that was focused on ecological effects of the parasites as breeds of sets or population dynamics. Most of them was carried in penguins, and most of them was carried in the subantarctic region. So we have a lot of work to do, and this is what we are trying in the last year to obtain, to get more information on this topic. And this is important because the disease is arriving to Antarctica. So it's a place very remote and something like that, but the diseases are arriving. We found in this last season this new disease present for the first time in Antarctic penguin. In this case, this is the Adelie penguin. This is a disease that has been found in 2006 and 2007. I remember in Magellan penguin and South African penguin, but never has been found at the moment until now in Antarctica. And so what the thing is, it's important in this case, this first record deserves the attention of the Science magazine, and spread this news in his portal of news in the internet. So it's an important thing. Moreover, in the last SCAR horizon scan that has, as you know, has developed the prospective research for the next 20 years. One of the questions that has been considered more important together with others is that how will climate change affect the risk of breeding and infectious diseases in Antarctica? But to have to respond to answer this question, first we have to know what diseases are native in Antarctica, to know what is new and which effect has. So this is the work we have to develop in the next year. So another example that illustrating the lack of information, the lack of knowledge we have about these organisms in Antarctica is ectoparasites, is ticks. In this case, Ixodes. Until 2011 that we published this paper about the distribution of the ticks in the Antarctic Peninsula, the only place that people know that the ticks was present was close to in the island close to the Palmer Station, in the Palmer Archipelago. There was a group of American researchers that were studying these ticks from the physiological point of view, to know how the ticks were adapted to the cold environments in Antarctica, with some surprising results. For instance, it seems that the ticks in Antarctica has 5 years life cycles, and that they adapted for some special conditions. But nobody knows if there are, or at least nobody published anything about the presence of ticks in other places in Antarctica. I must say that it's very difficult to find the ticks on the penguins. This is one of the reasons that most of the people thought that the ticks are not present, spread on all at least the Antarctic Peninsula. As an example, we are working in the Exception Island for the last 25 years. We have been there, a group of around 45 people in these 25 years, and nobody has been detected the ticks in in this time. So, where is then the ticks? The ticks can be found below the stones that are close to the penguin colony. You can see here the penguin colony. You can see this stone. You can see this is what you can see below the stone. You can see the eggs. You can see the males. You can see the nymphs. You can see the grand females. And in some places, I mean below the stone, you can see thousands of ticks. So they are there, and they probably cause some effects on the penguin. But as I mentioned, nobody knew nothing about this until 2010, 2011. Moreover, the distribution of the ticks in the peninsula in north of Linus, there is a gradient from north to south, being more abundance in the north and south. So this gives a scenario of colonization from the north to the south. The most expectable answer to this pattern, and to try to test if this is to confirm this expectation, we did genetic analysis using microsatellites of the ticks, and we found another surprise. Instead of one population in all the Antarctic Peninsula, we found two different genetically populations, one in the north and one in the south. The north fits very well with the expectation of colonization from the north. There is an hybrid place that is just located in the Section Island. We don't know how can it happen, or we don't know any answer, any explanation for this. But the most strange thing, or the more of a mystery, is how this population has been here, how has been genetically differentiated for this other population of pigs. We don't know if there is some colonization from southern places, but it's difficult to think. But we work to have more data, to form more ideas, and more hypotheses to be tested. But this is an example of the lack of information we have on these organisms in Antarctica. So the ticks transmit some disease and and they are vector of some other parasites. And we found, as expected, that the penguins show this bacteria, Borrelia tuberculare, that is responsible of the Lyme disease, and is present in all the populations. Even in the population, we have not detected ticks. This is the more southern population we have, in just south of the Adelaide Island. There are some contact with the Borrelia, so there are some ticks there, but we were not able to detect it. In fact, American researchers told me that there are ticks for years, so there are some ticks in 1 year that disappear the years after. And the other thing is that the ticks transmit this blood parasite, the Babesia, that is this one. We have the microscopic data as this, and we have confirmed by molecular techniques. So this means that the ticks are causing directly effect probably by eating the blood, but also by transmitting disease and other parasites. So we try to get as much information of all kinds of pathogens and parasites. This time we describe, or we try to look at the presence of bacteria and virus. This is some of the bacteria, the pathogenical bacteria we found. We did also phylosequencing, and we extend the presence of the number of bacteria to 3,000 different types of bacteria. And we also found that there are differences between adults and chicks. The chicks show less number of bacteria that the adults, as expected, but there are not many published information on this, not on penguins, but on wild birds in everywhere. So this is a first step to know what the penguins can manage with these organisms. Another important thing is to know not what organisms of parasites are present, but what organisms of parasites are absent, because in this case we show the absence of this protozoa, Cryptosporidium germ and Toxoplasma. And it's important to know, because if we detect in the future the presence of these parasites, we have a baseline for the time when these organisms were present, and we can explain how they arrive, and just in cases carried by carried by human to propose protection measures or code of conduct or something like that. More protozoa, the Eimeria, which is the intestinal protozoa. They are present in most of the populations. Also, it is important, as I mentioned before, to have data about the prevalence. The prevalence can change in a subtle way. In this case, it's linked with the probability of transmission in relation to the aggregation, to the colony size. The prevalence in this kind of colonies was of 30%, while in the most small colony, the prevalence was very low. And this will have also consequences for the immune system in the birds living there in comparison with the birds living here. We know very few about other parasites like the gastrointestinal parasites. In this case, for instance, this species is the first record for this species in the penguins. And so there were no information about this parasite. Now there are in some places, but there are many other places that we don't know if these parasites are present, or there are new species affecting, or there. Of course, we don't know nothing about the changes in the abundance of these parasites in different places. So there are, again, a lot of work to do. And at the same time, we have also the opportunity to study the parasites themselves. And in this case, we did some molecular characterization. First, to be sure of the identification of the parasites. We developed some molecular probes, because we have had some problems for detecting the parasites in the feces of the penguins. So we hope that this new technique can help us to increase the accuracy of the potential of the identification of the parasites, and to have more information. But also we have the opportunity, but it doesn't matter, this is not for reading, but it's just to illustrate, we have the opportunity to have obtain information about the biology of the parasites itself, I mentioned before, and to know which are the phylogenetical relationships between the parasite. This is what is our part. So to know to have information about the phylogenetical relationship between among the different parasite species. As an illustration, for some of the groups, we found just in a group that can be hundreds of parasites, we just found in the database, in the gene bank, information for 10 or for 5 to 10 different species. So again, we have a lot of work to do in this aspect. And the other thing we don't know nothing about is the life cycles of the parasite. We don't know nothing about the host, intermediate host, or the parasitic host. In this case, what we are working on trying to find the presence of the parasites in the krill. We found at the moment the juvenile larvae of the nematode in the krill, but in the very few individuals, so we have to increase the work on this to know how this has to know how they are transmitting and incorporating to the or infecting, incorporating to the virus. Another important thing is to know the effects of the parasite, of course, not just the presence of, but this is the most important thing, to know the effects. To work on this, to do this study, we use a technique that use anti-augmenting drugs. So we eliminate the parasites of the experimental group. We use, for close comparison, nest with 2 chicks. One of the chicks was injected with the anti-augmenting drug, and the other was injected with the placebo, to avoid another secondary variable that can confound the results. And we obtained that, as expected, that the experimental group, the experimental individual without parasite or in which the parasite was decreased, grow more, have more, at the end, more body mass than the control one that has the parasite that usually they have. I must say that the prevalence of this kind of parasites in the penguins are around 87%, so most of them are infected with these parasites. And we can estimate also that the reduction, that the effects of the parasites in relation with the body mass is around a reduction of 200 grams on average, which represents 6% of the body mass. We replicate this work with the gentle penguin. We obtained the same results. In this case, we also were interested in how the parasites can affect the presence of the carotenoid concentration. We will talk on the carotenoid a few minutes later. Because some of the parasites can take the carotenoid that are taken by the penguins, and in this case, the carotenoid are the compounds that give color to the red beak of the gentle penguin. In this case, we didn't find any difference. Can be explained because these parasites they don't take the carotenoids from the penguins. Can be explained because the penguins has enough carotenoids from the diet, and so the intake of their parasites is not affected. But, this is the results. To see the effects of the ticks, coming back to the ticks, we take advantage that in our place. This is the penguin property of Barbosa with section item, which is penguin. And there are differences in the places in the density of the ticks. These places, this colony have a high density ticks in the stones in the colony, and here there are very low density. Mainly the explanation mainly because here there are a lot of stone, while here there are very few. So the ticks are living below the stone, so there are few. And we found, sorry because it's in Spanish, but we found that there is more mortality of the chicks in the place with the high density of ticks. So this is a clear effect of mortality, but we didn't find any effect on the size of the chicks. This can be explained because in this case we work with the chicks at 20 days old, and the mortality was happened before, and probably we work with the penguins. For sure, we work with the penguins that survive, and there are the high quality chicks, and probably this is the reason that we didn't find any positive result. So we go to the immunoresponse. Very briefly, the immunoresponse is the first defense that the organism has against the effects of the parasite compared to the pathogens. There are different compounds. There are cellular immunity, mural immunity, and that I will not give explanation of this. Immunoresponse immunity can be affected by different factors that can produce immunodepression and reduction of immunocompetence. And so we have to be aware of what these other factors are affecting the immune response to be sure to know what are the effects of these kind of things on the, to explain the population decline that we are our final goal. We found that there are variability in different populations of the different species from north to south, from north to south, south to north, being more higher the immune response in the northern population that is fit with the presence of more parasites and more species and abundance of parasites in the region of the Antarctic Peninsula. This is the case also for most of the species with the cellular immune response. So we have that there are things that happen because of the parasites are affecting the penguins. We found in relation with, remember the differences that we found in the prevalence of the Imeria, but also for sure in the prevalence of different pathogens linked with the high probability of transmission in the big colonies. We found that the cellular immunity was higher expected in the more infected places. And we have also to know how the immune function has been related with other physiological functions, mainly highly energetic demanding, like let's say breeding of the molting. You know that birds, penguins, has to face with a lot of work during this phase of the life cycle, because they have to go to sea, to take food, to bring to the chicks, and again during almost 2 months and a half. But also the molting is a very energetic, demanding activity, because they lost all the feathers in 15 years, in a week more or less, and they have to grow the feathers at the same time. It's very different with the molting of most of the bird that is changing the feathers from time to time, so they have to face with the increase of metabolism of this. And we found that the cellular immunity, the immunity is different between both demanding energetical situations. For instance, it seems that the molting is energetically more costly than breeding, for instance, in terms of the effects on immunity. And in this case, the immunity has been decreased. So it seems in this case it can be a trade-off between molting and cellular immunity. And this is the same for the humoral immunity. So it seems that in this case, the penguin has to deal with the three demanding functions: the immunity, the breeding, and the molting, and have to put energy in a different function depending on the situation and depending on the problem. That means that if these birds are with the immune system depressed, if in this time there are an infection, it will be more, will have more risk to be dead or to be in a very bad condition. Another function that's important for the penguins in these cold environments are thermoregulation. We try also to find a trade-off between the immune system and the thermoregulation activity. But what we found it was the opposite. We found that the penguins show a positive relation between the thermoregulation effort, measure up the difference between the global temperature and the temperature in the air, with their immune response. So in this case, we mean that there is not a trade-off, and the high-quality individuals that can do a high thermoregulatory effort can at the same time have a high immune response. So this is a surprise. This is a process we have tested, and this is an illustration what we have to do this kind of work to know what is happening with the physiology of the penguins, and not to do some predictions without to be tested. We continue with now with the carotenoids. And well, it's important, the carotenoid. The carotenoid has are antioxidants compounds. In this case, the krill. The krill is it's a reservoir of carotenoids. It has a lot of amount of carotenoids inside, and then the penguin, when they take the krill has taken the carotenoid, it can be an explanation. For instance, going back to the talk of Michael this morning, why the penguin changed from the feces to the krill when the krill are available. It's an explanation because they are reward of taking this prey. And other explanation as well, of course. And so this is important for the carotenoids. Go back for fighting against the free radicals that it provoke in the activity and the many other situations, and the intake of for instance, the contaminants that increase the presence of the free radicals in the body. So the carotenoids and other antioxidants, for instance, the vitamin E or something like that, are also important. And it has important consequences in the secondary sexual character that we will see in a moment. And I have to say that the important thing with the carotenoids is that they cannot be synthesized by the penguins, by any animal. What they are probably a small acid that can synthesize, but it's not the usual situation. Animals have to incorporate the carotenoid by means of the food. So this is important that if the food is changing, and there are some changes in the diet, probably the carotenoid intake will be lower if the penguin increase the intake of fishes or squid. So as a consequences of the low intake of carotenoid, penguins will have problems with the eradication of free radical and other emotions. So to see how the carotenoids is functioning in the penguins, we first try to know how the variability of the carotenoids in the different individuals, in the different groups, and the different populations, and we found that there are differences in the breeding birds, the molting birds, and the chicks. The chicks show the highest concentration of carotenoids because they are fed by almost exclusively by krill, but the adults also intake other prey, so the level of carotenoid are lower, and they have to use also with other physiological functions. But the more surprise is that during molting the carotenoid disappear on the blood of the penguin because they stop to eat for 15 days before molting, but also because they probably use the carotenoids to fight against the free radical producing during the molting, and they cannot replace this use of the carotenoid by foraging. We also found that there are differences in the different populations. So it's clear that the penguin forages mainly on krill, but there are some differences in the different populations in the diet, as shown this morning, that affect the variation of the amount of carotenoid present in the blood. So this means that variations in the krill, variations in the food at sea, will affect this different concentration of carotenoids, and they will have physiological effects as a consequence. Relating with the secondary sexual characters, most of this kind of traits that show the birds and other animals, the color of their feathers and the color of the beaks, are the secondary sexual characters that use the male to express the quality to be selected by the female. In this case, in the giant penguin, there are no any demonstration of this mate selection process because it's very difficult at the moment. But we have one of the indicators of that this trait can be a secondary sexual character, and it's the relation with the body condition of the male. In this case, there is a clear positive relationship between the body condition of the males and the increase of the red of the beak. So the redder the beak is in the males, the more body mass, the better condition they have. There are also, we saw before that there is differences in carotenoids between population and population, and we also found differences in the color of the beak in different population, reflecting these differences in the carotenoid present in the different individuals. Yes, these differences can be explained because of the ability of the availability of the krill. In this case, we know that the krill is larger in offshore environments than in inshore, and therefore there would be more astaxanthin in these places than inshore. And using stable isotopes, in this case carbon-13, as explained Mike this morning, that reflects different habitats, differences in offshore foraging and inshore foraging. We see that it fits with the difference in the carotenoid, in the color beak. So it means that these differences in the places and the difference that is related with the quality of the krill that they are intaking affecting other traits like the beak color. The beak color, it's actually trait like it's selected by sexual selection. Changes in the diet, changes that can be in the carotenoids, and changes that can appear in the color of the beak could be evolutionary consequences of very long term, or at least changes in the process of sexual selection in this population. And this is a very unexpected consequence of the climate change. Remember that we are talking about the climate change at the beginning and consequences at the end, so this can be a consequence for this process. And finally, there are human interactions different from the climate change. There are contaminations that are produced by humans, human activity. There are also human disturbance, and that is this factor affect the immunodepression. We seen yesterday a very good talk about this relation between contaminants and immunity, and this is a kind of things that illustrate the sublethal effects that some of the human activity can affect the penguin. There are a lot of there are some works that are trying to see how the human visitation, for instance, has been affected the population of the penguins. There are different results, so there are disagreements. Sometimes because of the methodology, sometimes because the researchers has just pay attention to the breeding success. But in this case, there are clear sublevel effects mediated by the physiology, and this is one of the importance that we have studied the physiology for explaining what happened to the penguins, to the population of the penguins. In this case, we did just describe the presence of different contaminants. In this case, are heavy metals, increased from north to south and very linked with the presence of the human, with visitation of the presence of the research stations. We also show that there were the identification of the accumulation of these metals in the penguins. That we have very interesting information to know how these contaminants are remaining in the food web. We also study the presence of organic pollutants in the station of penguin to see how this is even more interesting than the metals, because the metals, there are human origin, but they are natural origin, and so sometimes there can be these confounding effects, but in the case of organic pollutant, it's clear that the origin is for human activity, and it's very important to know which kind of pollutants, of organic pollutants, are present in the penguins and the effects of the physiology. And I am very happy to know that there are a couple of posters dealing with this information, because it's the same, is similar with the information of that you have very little information about the presence of contaminants, and of course very little about the effect of the contaminant on the animals. And finally, the direct human effects. This case is represented by the tourism, but can be also the effect of the researcher or a kind of visitation. And in this case, where we try to do was, well, this is just to illustrate the increase in the last times in the number of people visiting Antarctica. So it's also we give some information in this talk. But what we tried to do was to combine all the things we are working in the different parts of the graph we have seen, to combine to see how to have a battery of indicators of how the human visitation or the human activity can be affecting the penguin. Well, this is in Spanish, sorry. We study the contaminants, the heavy metals, the erythrocyte malformations, this kind of things that you can see in the erythrocyte in the blood. The usual nucleus of the erythrocyte is elliptical, and you can see this different characteristics. The micronucleus is one of the most common malformations that you can find in the blood of the animals. We measure also the stress level. We measure the presence of bacteria transmitted by the human, like Campylobacter dysunic, which is the transmission by the human, versus Campylobacter lari, that is the endemic of the birds. We measure also the level of stress and other parameters, immunological parameters. We didn't find any different in this bacteria, so it's a good news, but there are not any Campylobacter dysunic introduced by the human in these places. I had said that the places chosen for the study was Hannah Point, that is one of the more visited places in the Antarctic Peninsula, with a maximum of 3,477 people visiting the place in 2005. There is lower in the last time because of the crisis. That is a very highly visited place. And we compared it with this penguin rockery in Bayes Peninsula, which almost nobody has visited in the last 50 years. Or very few people. And we found that there is higher contamination of heavy metal, for instance, lead in the more visited penguin rockery. There are higher genotoxic damage that is expressed by the presence of erythrocyte malformations. There are also immunological changes. There are also a surprising finding, because we found that the stress was higher lower in the more visited penguin rockery, and the expectation was of the opposite. And there are some other studies dealing with the presence of people in other penguin rockeries, in Magellanic penguins and other kind of animals, that has shown that the continuous visitation of people of these places or these animals provoke an habituation of the animals that lower the response of the stress, because you know the response of the stress is not very good for the body. There are many negative influences of other physiological things. So animals cannot maintain a high level of stress response, and because of the visitation, they are habituated to people, and they lower the stress response. So this is an indication of the habituation of this place, and it's illustration of the effects of the hemophilia here. And I think that this is the end. So I try to demonstrate, or to show you that all these different things, there are, I say it again, a lot of work to do to actually demonstrate how it's the effects of the different factors in order of the cascade effects in the different steps of this graph. But I think this is a good proposal for working, have idea for formulate hypotheses and predictions to work on this way, and finally to try to explain the population declines and other kinds of things. As I mentioned before, populational changes are just the tip of the iceberg that we can see on the surface of the water, and there are many other different factors. At the moment, hidden factor, that is the physiological changes. You can imagine all this kind of data that is just a small piece of result we have. We have a lot of more results in another factor. It's thanks to the work of many different people in the field, and also in the lab. We collaborate with many different institutions that has researchers specialized in different things. I just coordinate the things and do something, but I know, of course, yes, I have not the, of course, the expertise for all of these kind of things. And I think that's all. I took it up.
Thank you very much, Dr. Barbosa, and we have some questions for you. We have time for 2 questions.
Two quick questions, Andrés. The first one is if you already addressed the potential role of birds wintering outside Antarctica in transporting pathogens, parasites to Antarctica and the penguins. And the second one, but related to the first one, is if could exist any role of rehabilitation centers outside Antarctica in